You ever hear about microRNA? It's wild. We're diving deep into these tiny things, and let me tell you, they're controlling, like, everything. How we grow, diseases, you name it.
Yeah, these microRNAs, they're fascinating. Just these tiny molecules, right? Only like 21 to 23 nucleotides long.
21 to 23, wow, that's small.
And yet they're like master regulators, flipping genes on and off, controlling which proteins get made. It all boils down to gene expression. They're running the show.
Okay, so for those tuning in who might not be scientists, could you break down what microRNAs are exactly? Like, what do they actually look like?
So imagine like a recipe, but super complex, right? Tons of ingredients. You need the perfect amount of everything at just the right moment to bake a cake. Yeah. That's what microRNAs do for our cells.
So they're like little chefs in our cells, making sure all the ingredients are right.
Exactly. They're these little chefs making sure the right genes are expressed when they need to be. And they're everywhere. Plants, animals, even viruses. It's crazy.
So we've got these tiny molecules controlling our genes. But how do we even discover them in the first place?
Well, it all started in '93 with a tiny worm. C. elegans, they call them. Scientists were looking at how it develops, and boom, they found this little RNA molecule called LIN-4.
LIN-4.
And get this, LIN-4 was controlling when the worm hit its developmental milestones, like when it molted or whatever.
Oh, so before LIN-4, no one knew that RNA could be directly calling the shots on gene regulation.
Nope. Total game changer. Back then, everyone thought proteins were the big bosses of gene expression. RNA. Just a messenger, right?
Yeah.
Carrying instructions. But LIN-4, it showed that RNA could be in control.
So basically, LIN-4 blew the lid off what we thought we knew about RNA.
Pretty much. And then in 2000, boom, another discovery. LET-7. Another small RNA also involved in development. But here's the kicker. LET-7 wasn't just in this one worm. It was in tons of species, even us.
Whoa. So not just some fluke in a worm, but something way bigger going on.
Exactly. And that's what led scientists to this idea. Maybe there's a whole family of these little regulatory RNAs. And that, my friend, is how the term microRNA was born.
MicroRNA. It's got a ring to it, doesn't it? But hold on, we got to talk about how these things are actually made, right? Because they don't just pop out of thin air, do they?
Oh, it's way more interesting than that. It's like a whole microscopic manufacturing process. So you've got DNA, the blueprint of life, right? And then there's this enzyme called RNA polymerase II. And that enzyme, it transcribes the microRNA sequence from DNA into this longer precursor molecule. We're talking primRNA.
primRNA. Okay, so that's like the rough draft.
Yeah.
Before it's ready to go out there and, regulate genes.
You got it. Think of it like this. The primRNA, it still needs some work. It's got to go through this whole refinement process, right? Like a molecular factory, basically.
That's factory. Inside our cells.
Exactly. So first, it's inside the cell nucleus, and this enzyme complex called the microprocessor gets to work.
The microprocessor.
Yeah.
Sounds kind of intense.
It is. It's a big deal. And the result, we get this shorter, hairpin-shaped structure. That's our primRNA.
So we're like refining the instructions.
Exactly. Making them more concise, elegant, you could say. But it's not over yet. This primRNA, it's got to move out of the nucleus into the cytoplasm. And that's where it meets the dicer enzyme.
Dicer. That doesn't sound very friendly.
Let's just say dicer's got a very important job to do. It's like a molecular tailor, see? It cuts the primRNA very precisely, and out pops this short, double-stranded RNA molecule.
Trimming it down even further. Getting serious now. So what happens to this double-stranded RNA thing?
This is where things get really cool. One strand from that double-stranded RNA molecule, it gets chosen, and then it's carefully guided into this protein complex called RISC.
RISC. Okay, now we're talking acronyms. What's that stand for?
RNA-induced silencing complex.
Silencing, huh? Sounds kind of ominous.
Well, silencing is the name of the game here. Remember how we're talking about controlling gene expression?
Right, like turning the volume down on certain genes.
Exactly. So this RSC complex, think of it as the microRNA's secret agent partner. They team up, see? And that mature microRNA, using its unique sequence, it finds its target mRNA, that is, and binds to it. And that's when it gets to work, silencing that gene.
Wow. That's a whole lot of action for something so tiny. So it's like the microRNA is programmed to find its target and shut it down.
That's a great way to put it. This whole journey from DNA to RISC, it just shows how incredibly complex and precise gene regulation really is. But there's even more to the story. Remember how we talked about microRNAs silencing genes by targeting these mRNAs?
Yeah, like what's the actual mechanism? How do they actually, silence the gene.
Okay, so it all comes down to something called sequence complementarity. Basically, microRNAs, they target mRNAs by binding to complementary sequences on them. Imagine a lock and key, right? The microRNA, it's the key,
Right.
and its target sequence on the mRNA, that's the lock.
So, like, if the microRNA sequence matches a sequence on the mRNA, they click together.
Exactly. And in animals, these microRNAs, they usually target sequences in the 3' UTR and the mRNA. That's an untranslated region, by the way.
Untranslated region. Got it. So what happens when they bind together? Does it, like, just destroy the mRNA?
It can. One possibility is that the mRNA gets degraded, like chopped up and destroyed, so it can't be translated into a protein.
No more instructions.
Exactly. The other possibility is that translation itself gets blocked. So the mRNA, it's still there, but it's like frozen. It can't be used to make a protein.
So the microRNA, it can either trash the instructions or just, like, lock them up so they can't be read. Pretty clever.
That's exactly it. That's how these little microRNAs are controlling gene expression.
Yeah.
They're regulating protein production with incredible precision. But here's the thing. This tiny mechanism, it's playing out on a much bigger stage, right? We're talking about our health, our behavior, everything.
Okay, now that's what I'm talking about. Give me some examples. How do these little regulators affect our everyday lives?
All right, let's talk about heart health. Studies have shown that in diseased human hearts, the expression of certain microRNAs, it actually changes. Some microRNAs, they're like the guardians of a healthy heart, while others, well, they might actually contribute to heart disease.
So some are the good guys and some are, like, working against us.
Exactly. Take microRNA 29, for example. Research suggests it could protect against heart failure.
Really?
Yeah. Scientists are super excited about this because it could lead to a whole new way to treat heart failure. Just imagine boosting microRNA 29 levels to protect the heart.
Wow, that's amazing. So by understanding these tiny molecules, we could develop groundbreaking therapies for heart disease. What about other diseases? What about cancer?
MicroRNAs are heavily involved in cancer as well. Interestingly, the first human disease linked to microRNA dysregulation was chronic lymphocytic leukemia.
Leukemia.
Yeah. And it turns out that a lot of microRNAs, they actually play a role in how cancer develops. Some act like tumor suppressors.
The good guys.
Right, and others act like oncogenes.
The bad guys.
You got it. And because they can influence cancer development, they're now seen as potential therapeutic targets.
So we could actually control cancer by, like, tweaking these microRNA levels.
That's the idea. Scientists are really excited about the potential for developing new cancer treatments that work by manipulating specific microRNAs.
That's incredible. It's amazing to think that something so small could have such a huge impact. And it's not just diseases, right? You mentioned microRNAs even affect things like our behavior.
Oh, absolutely. Let's take alcoholism, for example. Chronic alcohol abuse, it actually changes how genes are expressed in the brain.
So alcohol can, like, rewire our brains at the genetic level.
In a way, yeah. It's more like it's changing the instructions for how our brain cells work. And a lot of microRNAs, they're affected by alcohol abuse. Some of them might even play a role in how someone goes from just drinking casually to becoming dependent.
So microRNAs could be involved in an addiction. That's fascinating. It's like a whole other level of control that we never even knew about.
It really is. And the more we learn about microRNAs, the more we realize just how much they influence. I mean, they're involved in pretty much every aspect of our biology.
It's like discovering this whole hidden world inside our cells with these tiny molecules running the show.
That's a great way to put it.
So for those of us who aren't scientists, why should we care about microRNAs?
Well, for one, they've completely changed how we think about gene regulation. They've shown us that these really complex processes can be controlled by these tiny, elegant molecules. And because they're involved in so many different diseases, understanding them could be the key to developing new, more effective treatments.
It's like opening up a whole new frontier in medicine.
Exactly. And the research, it's moving so fast. Every day we're learning more and more about microRNAs, and the potential they hold is just incredible.
It really feels like we're on the verge of a new era in medicine. Well, this deep dive has been fascinating. I've learned so much about microRNAs, and I hope everyone listening has too.
It's been my pleasure.
And all our listeners, what are your thoughts on microRNAs? How do you think this research might change the future of medicine? We'd love to hear from you. And with that, we'll wrap up this deep dive into the world of microRNA. Thanks for joining us.